Hello everyone, welcome to the last lecture of Automation of Scientific Research. My name is Chidi. Today I'm representing my team to give the lecture related to in vitro protein production optimization. Here is a brief outline for today. First, we'll go over some backgrounds of cell-free protein synthesis. Next, we will discuss today's paper, Large-Scale Active Learning Guided Exploration for In Vitro Protein Production Optimization. First, here are some backgrounds on cell-free protein synthesis. It is also called in vitro protein synthesis. It is a biological technique that produces protein using biological machinery without the use of living cells. There are a bunch of advantages to cell-free protein synthesis. First, it is much faster because it does not require gene transfection, cell culture, or extensive protein purification. So it usually takes 1 to 2 days to finish up the experiment. In contrast, as for in vivo protein synthesis, which means producing protein within living cells, it usually takes 1 to 2 weeks. Second, cell-free protein synthesis is an open reaction, so it is easy to be monitored. The lack of cell wall allows direct manipulation of chemical environment. Samples can be easily taken, concentrations can be optimized, and the reaction can be monitored. In contrast, as for in vivo protein synthesis, once DNA is inserted into living cells, the reaction cannot be accessed until it is over and the cells are lysed. Another advantage of cell-free protein synthesis is that the lack of concern of toxicity. Some desired proteins are toxic to cells when synthesized. Since living cells are not being used, the toxicity of the product's protein is not our major concern. Here's a one-slide summary of the paper. The goal is to optimize buffer composition to maximize protein production with minimal experimental effort. The data access model used is membership query synthesis. The query selection strategy is to maximize upper confidence bond, UCB. Now let's dive into the paper. First, I want to discuss the search space of cell-free buffer composition. As we just mentioned, the goal is to optimize buffer composition to maximize protein production. Inside of the buffer solution, there are 11 chemicals. 4 concentrations have been chosen for each chemical. For example, the first component is called magnesium glutamate, and the 4 concentrations they chose are 0.4, 1.2, 2, and 4. In total, the combinatorial space is 4 to the 11th because we have 11 chemicals and there are 4 concentrations each, which lead to 4 million compositions, which is a huge search space. And now we're ready to enter the active learning loop. In order to initialize the base learners, the researchers need to have some training data set. They designed 102 initial compositions, which includes 22 concentrations they wish to test and 80 random concentrations. As for these 22 concentrations they wish to test, these include they first fixed all reagents at the maximum allowed concentration, except one which was at the lowest, so this led to 11 combinations. Reversely, they also fixed all reagents at the minimum allowed concentration, except one which was at the highest. This led to another 11 combinations. And after they designed the compositions, they're ready to take the protein production measurement. For each composition design, the researchers put 11 chemicals with the specified concentrations into a test tube, together with a well-designed plasmid. The plasmid carries a promoter and a GFP gene. Therefore, protein production can be measured using the fluorescence level from the expression of the GFP gene. In the middle column of the figure, it shows 5 different compositions and the corresponding fluorescence level. For example, as for the reference has all chemicals at the highest concentrations, and it has the lowest fluorescent level, which means a lowest protein production. On the right-hand side of the figure, there is a plot of yield versus composition. To be noticed, reference has a yield of 1 because the yield is calculated as the fluorescence of the current composition minus autofluorescence divided by the fluorescence of reference composition minus autofluorescence. The autofluorescence can be viewed as a background. As shown by this plot, different compositions result to different yield.
After they had the initial training set with 102 compositions, they were able to initiate the base learners. They trained an ensemble of 25 models. Each model is a multilayer perceptron, and the multilayer perceptron was initially initialized by random weights and trained with maximum iteration of 2000, adaptive learning rate, atom solver, and 4 hidden layers. Actually, they trained each of the 25 models 10 times and kept the best model of the 10 models for optimization. After they train an ensemble of 25 models, it needs to predict the best compositions to test for the following round. Here comes to the query selection strategy, upper confidence band maximization. It is able to balance between exploitation and exploration, as we will see next. Here is a pseudocode for the query selection strategy. The iteration time was set to 100,000. They randomly sample a composition in the composition space. If a composition was joined previously, either in a previous experiment or during current selection, they simply neglect it. Otherwise, they predict mean and standard deviation using the ensemble of 25 models previously trained. So at the end of the iteration loop, we would expect to see 100,000 compositions and its corresponding mean and standard deviation of the predicted yield. In order to select the best set of compositions, they use this upper confidence band formula, which is exploitation times the mean of the predicted yield plus exploration times the standard deviation of the predicted yield. The exploitation was set up to 1 and exploration was set to square root of 2. Here, a high standard deviation value stands for an uncertain yield value. They therefore queries point with both high yield and uncertainty. Their script outputs the best 500 compositions. After they chose the compositions for next round, they are ready to do the wet lab experiment. As for each round, 102 new buffer compositions were tested. These 102 new buffer compositions was extracted from the best 500 compositions in the previous step. They made 3 replicates for each composition. After they get the yield for the new buffer compositions, they updated the model using the whole dataset so far, and the loop continues. Now let's look into some results. First result is related to the yield evolution and prediction accuracy among 10 iterations. Here in figure A, each column represents the result from 1 iteration. The green dot represents mean yield of the 3 replicates, and some little gray bar represents standard deviation of the 3 replicates. As we see in the first iteration, they already achieved a 10-fold improvement of yield. However, the prediction accuracy was really low here. After 7 iterations, they reached a maximum for both the yield and the prediction accuracy here. The yield was 34-fold and the accuracy, the R-squared value is 0.93. Eventually, they stopped at the 10th iterations as they were not able to increase neither the yield nor the prediction accuracy of the model. They also analyzed the relationship between chemical compositions and yield. Each column here represents a composition. The yield of the compositions are ranked from highest to lowest, and the rightmost column here is the composition that produces highest yield. We can observe some patterns by checking each rows here. Red color means a high concentration and green color means a low concentration. As we see, an increase of magnesium glutamate, calcium glutamate, amino acid, and NTP's concentrations, and the decrease of CAMP, spermidine, and 3-PGA concentrations can be noticed with increasing yield. Third, they generated a one-step method to predict protein yield. This step is to check whether protein production in cell-free system using lysis made in other conditions could quickly predict it with a one-step method. This example shown here is the result of lysis made by a different experimentalist. The one-step method started with selecting 102 composition representatives of the 1,017 compositions already tested with the original lysate. Among the 102 compositions, they split them up into training and testing datasets. 20 were used to train the model and 82 to test its predictive accuracy. To be noticed, the 20 here were used to train the model were not chosen at random. The choice of the 20 buffer combinations actually led to the best predictions. See the paper for details. As shown by the middle plot, the R-squared were computed on all points and Q-squared was computed on 82 points of the test set. Both of them achieved 0.93, which is a really high accuracy. Therefore, they concluded that their approach is able to accurately predict protein production using a small training set, regardless of the experimentalist who prepared the lysate. Here is a summary for today's lecture. The authors used active learning strategy to optimize cell-free buffer composition among a space of 4 million buffer compositions, maximizing the protein yield. The researchers measured fluorescence level in 1,071 cell-free buffer compositions, and the model achieved a high prediction accuracy, R-squared equals 0.93, and a maximum of 34-fold increase of the yield. Third, chemical components critical to cell reproductivity were identified. And here are our references. Thank you for your attention.